after the coffee break. I'm delighted to introduce our next speaker, Dr. Emma Dempster. She's a lecturer at the University of Exeter Medical School, where she's studying epigenetics of mental illness and other complex diseases. Her current research includes a wide range of projects, including genome methylation in monozygotic twins who are discordant for schizophrenia, epigenetic signatures of genome deletions, and so on. She finished her PhD in molecular genetics of schizophrenia at the Institute of Psychiatry, King's College London, where she studied genes involved in brain development and their role in schizophrenia. Today she'll be talking to us on epigenetic analysis on schizophrenia. Emma, the floor is yours.
Thank you very much, and thank you for putting on this amazing day today. So I'm going to talk about some of the work that's been going on in our lab, in particular three projects involving schizophrenia. So the first, which John's already sort of mentioned and sort of stole my thunder a little bit, is the epigenetic analysis of twins discordant for schizophrenia. The second project is again an epigenetic analysis of 22q11 deletion syndrome, which is a deletion some of you are aware is strongly associated with an increased risk to schizophrenia. And then the third study I'm going to talk about today is the functional analysis of a schizophrenia GWAS and EWAS candidate gene, and some of the methods that our lab is currently undertaking to sort of look at the functional consequence of some of these both genetic and epigenetic changes in schizophrenia. So this is the first study. All of this work I'm going to talk today is pretty much unpublished. The twin study here I'm going to talk about now was in the first meta-analysis that John and myself and Eilish published a couple of years ago. So, as John's already mentioned, sort of why MZ discordant twins are so powerful for epigenetic analysis is that they share the same DNA, they have the same parents, usually they're the same age when you profile them, same sex, same room, and early life in home environment. And as John's already mentioned too, that sort of common diseases are highly heritable, and particularly if you look at schizophrenia, which is, get the laser pointer. Schizophrenia here, when you look at MZ twin concordance levels, they're around 50%. So, which obviously suggests it's highly genetic, which is why I've been, since my PhD in 2005, been looking at the genetics of schizophrenia. But also, you've got to think the sort of the flip side of the coin is that these identical twins are also different. They're discordant for schizophrenia. So, if your identical twin has schizophrenia, you have, a 50-50 chance of also developing the disorder. And it's that difference that we really want to look at when we're looking at epigenetics. So, as I mentioned before, disease-discordant MZ pairs are fully matched in early childhood effects, and particularly genetic factors. And also, studies of disease-discordant twin pairs have increased power compared to unmatched case control series. And this is a paper by Jordana Bell a few years ago looking at power analysis, just showing that the twin design is much more powerful than case controls when you're looking at an epigenetic analysis. So this is our sample set. So we have 96 MZ twin pair DNA samples, which the DNA was derived from whole blood. 75 of which are discordant for schizophrenia. And we had 7 pairs that were concordant, and then 15 controls, and pretty much equal sex ratio, and they're mostly middle-aged, around 40 years. So these samples are all Northern European, and they're from a number of centres. The Maudsley Twin Register in the UK, which is Professor Robin Murray started a number of years back, the Netherlands, Germany, Finland and Sweden. So we did the DNA methylation profiling, and we used the old array, which is the full 50k human methylation array. So this study was done a couple of years back. And then we used the same packages that I'm going to talk about tomorrow to do the normalisation and the QC, and I'll go into more detail tomorrow. So the data analysis, we did what we call a differential methylated positions. This is where we just take each individual probe to see if they're associated with the phenotype. And we did a paired analysis, so basically look at a paired t-test to see, if there's any difference between the affected and unaffected twin. And in this analysis, we were just looking at the discordant twins. We then did a more akin to a case control analysis. So we were able to incorporate the control twin pairs we had and also the concordant for schizophrenia twin pairs we had. And this we used a regression-based analysis, and we used robust, it's called robust standard errors. So we're able to factor in the fact that a lot of these twins were related. We also controlled for, in this analysis, age, sex, cell composition, smoking scores, and the batches. And we had 99 in that cohort. So this is the two sort of results tables here. The top one here is the paired analysis, and then the bottom one here is the sort of case control analysis here. And as you can see, the top probe from both analyses is in this gene HDAC4. So this is the Manhattan plot here, and as you can see, we only really saw one genome-wide or array-wide significant hit, and that was in HDAC4 for both analyses. So this is a plot just looking at the differences between each individual twin pair. So each little blue bar here is a twin pair looking at the differences, so difference between the DNA methylation, so what we call the beta differences. And as you can see, apart from a few that are going in the opposite direction, which we'll kind of ignore, the majority are all the changes in the same direction. And what we did was we looked in a large MZ twin cohort that was a population-wide, so they had no phenotype. And what we did was we sort of randomly assigned the twins case or control, just to really look at the variation between twins in a normal population, and we permuted this a number of times to see. And that's in the green there here. You can see the distribution of this, the DNA methylation changes within that the normal population of twins is what you'd expect a normal distribution to be. Where if you look in our schizophrenia discordant twin pairs in the red there, you can see that it's shifted there, and we see hypermethylation at this particular site in this gene in the discordant twins. So, this was obviously just one site in the genome here, and it's in an intronic region of a gene called HDAC4. So HDAC4 is a histone deacetylase. It's highly expressed in brain, and it's involved in synaptic plasticity and memory, as well as other neurological process. Also, histone deacetylase inhibitors have a long history of use in psychiatry and neurology as mood stabilizers, so it's obviously a really interesting candidate and an epigenetic candidate in its own right. So then we looked at some other data sets that John's already mentioned, that we've been profiling lots of different cohorts, schizophrenia cohorts, and this time we had access to two at UCL and both Aberdeen, both quite large blood-based DNA EWASs. And as you can see, the top twin probes, so we looked at 369 and as you see, 316 replicate within the same direction in the UCL data set, and then 312 in the Aberdeen, and both of these were highly significant if you did a sign test. We then just sort of looked at the most significant ones, so 125 of the top ranked 369 probes were significant in the same direction in the UCL replication cohort, and 74 were both significant in both cohorts. And these are just nice sort of scatter graphs here, which John's already sort of mentioned, where you can compare the beta differences between two different populations, and it's quite a powerful way of showing data, sort of replication data. And again, another way of showing sort of a replication data is this is what for the one particular CPG that was significant in HDAC4 in the twins. And you can see that this particular site replicates in both the UCL and the Aberdeen. So giving us real sure that although our twin study was much smaller, because obviously it was just 99 twin pairs compared to some of these big case control studies, which were nearly 1,000, you can see that we're getting reproducible signals, suggesting that it's a valid method. We also did a meta-analysis the three different, so using the top results from the twin analysis, and we found that HDAC4 and DISC1 came highly significant. Of course, DISC1 is a gene that's been associated with schizophrenia numerous times. We also looked at regions, so this is what we often call them DMRs, so this is differential methylated regions, and we use a program, it's a Python script called COMP, and this is basically looks at p-values, so it looks at the output from your analysis, and it looks to see if there's clusters of p-values, and then what we do, we'll go and then look at the demethylation data, and what we usually find is that they're in the same direction of effect. And from these analysis, we found 3 different regions that came up, all of which were on chromosome 6, 2 of which were close to or in the MHC region. And one of these regions did replicate in the second cohort. So this obviously could be interesting given the MHC has been associated with schizophrenia. So as John's already talked about, schizophrenia we know is polygenic. It's obviously there's going to be many genes of small effect. And there's you can't be in schizophrenia research and not heard about all the schizophrenia polygenic risk score. And currently it accounts for about 9% of the variance in caseness. And in the UK biobank sample, the schizophrenia polygenic risk score predicts lower performance in a variety of cognitive tests. But at the moment it's not diagnostic, but it is a useful tool in research. So we've looked in our cohorts that we've done our EWASs on. These are schizophrenia case controls. This is in both UCL and Aberdeen, and you can see that our cases have a higher polygenic risk score than the controls, as you'd hope if the polygenic risk score was working. But we were really interested to see what the twins look like, and well, John's kind of already stole my thunder there because he's already told you the answer. But we work because obviously they're identical twins, and one has schizophrenia, one doesn't, and what is their genetic risk? But actually, when we looked at them, they have the discordant twins have higher risk, the same as concordant twins. And we did a t-test comparing the schizophrenia discordant twins with those in control twins, and it was significant. You can see there, and the discordant twins are sort of the red here, and then you've got the concordant twins here, and you can see they're pretty much identical, and then these are the control twins here. There's obviously quite a small cohort as well, because you're comparing 70 with 30 control twins. And this is obviously very striking. So perhaps maybe we should be looking at when we're looking at discordant twins, we should be looking at the twin that doesn't have schizophrenia, and to see maybe what differences they have rather than the actual affected twin, because they obviously have the genetic predisposition to schizophrenia, but they haven't developed the disorder. So I'm going to sort of change tack slightly. So along with common genetic variation, the sort of big question in the schizophrenia community has been, can rare or de novo variation be contributing to schizophrenia? So one thing I'm particularly interested in is copy number variance, which is gains and losses of large chunks of DNA sequence. And it's usually defined as a segment of DNA that's about 1 kilobase or larger and present at variable copy numbers in comparison with a reference genome. It can encompass whole genes or important regulatory elements. And it involves far more nucleotides than SNP variation does. And it can be inherited, or it could be a de novo event. And multiple rare CMVs have been found to contribute to complex psychiatric disorders such as schizophrenia and autism. And this is just a figure here showing the different CMVs that have been associated to schizophrenia. As you can see, there's quite a few of them. And in particular, the most significant one is this one at the bottom here, which is 22q11, or I was told by cytogenetics you're supposed to say 22q11, but I'm going to probably call it 22q11 from now on because it's a habit. And as you can see, this is highly associated with schizophrenia. So the next... 22q11 2 deletion syndrome, or sometimes called DiGeorge syndrome or velocardiofacial syndrome. It was first described as a clinical condition in the 60s, but it's been known to be associated with schizophrenia since the early 1990s. And about 1 in every 4 individuals with this particular genetic deletion actually goes on to develop schizophrenia, or there's other psychiatric phenotypes as well associated with this particular deletion. And in unbiased prevalence studies, they do show that there's actually an estimate this deletion occurs in about 1 in 100 to 200 patients with schizophrenia. So this really is the first true molecular genetic subtype of schizophrenia. It's the closest we've got to, I guess, sort of a single gene, form of schizophrenia. But obviously, it's not a single gene; it's about 90. But and there's also, apart from schizophrenia, there's quite a few other sort of neuropsychiatric phenotypes of this particular deletion. There's neonatal seizures, often you get developmental delay and intellectual disabilities. There's autism spectrum features, ADHD, recurrent seizures and epilepsy, and some mood and anxiety disorders. Obviously schizophrenia, because that's why I'm talking about it here, and also some neurodegenerative disorders, particularly Parkinson's disease. The schizophrenia seems to be the same as it would. It meets the DSM criteria in patients with this deletion, apart from the median age of onset tends to be slightly higher at around 21 years. So it's usually it's a hemizygous deletion on chromosome 22q11 that means it's like heterozygous basically. So you just have one of your 22 chromosome 22s with this particular deletion. It's caused by non-allelic homologous recombination. You have these low copy repeats and basically you get mispairing at meiosis that causes this deletion. So it is quite common when you CMV. Most changes are de novo, so this is just a de novo event in the individual, but it has been found to be inherited from a parent. When there's a slight excess of maternal origin. There's about 90 genes in this region, and most are expressed in the brain, so that doesn't really help sort of rule out any particular other genes. And many, but not all, have decreased expression with the hemizygosity. So this is a study I've been doing with a postdoc in our lab called ESAN, what we did, we collected 64 22q11 deletion carriers, and this is from the Netherlands, and also we had 162 controls from the Netherlands also. We used the new Infinium methylation EPIC array, which I'll talk about more tomorrow, and we first of all just did a very simple 22q11 deletion patients versus controls, and then we also did within 22q11 deletion an analysis, because we had a number of individuals that had psychosis and a number of individuals that weren't exhibiting any psychiatric symptoms, to see if there's anything going on there. We looked at, so we did both what I talked about earlier, the same with the twin study. We looked at the individual probes, so the differential methylated position analysis and also the regional analysis. We're still this is work that's still ongoing at the moment. Essen's just become a father, so he's sort of disappeared back to the Netherlands for a while, so I'm hoping he'll come back soon to help me finish this work. But we also have we've genotyped these individuals as well, so we've run them all through a program called Penn CMV, so we have the CMV lengths and everything like that, so we want to sort of incorporate that into our analysis as well, and also calculate the polygenic risk score as well, to see what where these individuals fall in the sort of when you look at the polygenic burden of disease. So this is just the sample demographics. As I was saying, we've got 64 with the deletion, and we've also got in our control sample that we also have individuals with schizophrenia without the deletion as well, just so because we wanted to do various different comparisons. So this is our analysis here. So this is our, when we were just looking with individuals with the deletion compared to our other samples. So this is the model we ran here, and I'll talk everyone through this a bit more tomorrow. So this is a linear model here. So we've got DNA methylation, and this is the what we're interested in, whether the deletion itself is having an effect on DNA methylation, and we're controlling for psychosis, because obviously we have psychosis in our controls as well as deletion samples, age, sex, smoking school, batch, and different cell types that we've calculated from the denomethylation data. And as you can see, this is the Manhattan plot here. It hasn't come out too right, but we've got quite a few positions that reach Bonferroni significance, and we get this huge peak here on chromosome 22, which is within the deletion region, which we have actually seen previously with another CMV we were looking at, which is quite intriguing. But first of all, when we first saw this, we thought it was possibly like an artifact, and maybe it was to do with just not having as much DNA in that region. But what we do is we see the changes go in both directions. So we see an increase in denomethylation and a decrease in denomethylation, and it's not in all probes in this region. So we think there may be something real going on there, but we're still trying to tease that apart, and we're sort of doing more analysis at the moment in the lab to really sort of tease apart what these CMVs are doing to the denomethylation sort of gene regulation generally. So this is just like a heat map here of the top 100 probes that are associated with the deletion. You can see that it nicely separates the samples from the controls, and there's one little blip here that John pointed out. What's going on with that one there? I mean, I do know from quickly scanning through this CMV data that we got from Penn CMV that there is one individual that has a much smaller deletion than everyone else, so I'm kind of hoping that's that person there, but we'll see. And this is a volcano plot here of the most significant probes that reach Bonferroni significance. And as you can see, and most of you have been working on schizophrenia research for a while, like myself, know that this gene here has been a very popular candidate gene for many years, COMT, and that's obviously been associated. There's two different probes quite far apart. One you see a decrease in denomethylation, the other an increase. And you can see that there's obviously clusters, in particular genes in this region that seem to be affected with denomethylation. So we're currently trying to sort of investigate these genes a bit further and to see what's going on here. So then we sort of looked at the regions as well. That the top region that came out from this analysis was actually not on chromosome 22 but on chromosome 7, and this is a gene called HOXA2. And here it is sort of plotted out, so you can see that it spans a number of CPGs, all that show a decrease in denomethylation. Is that right? Yes. And what's interesting as well, that mice that homozygous for a targeted mutation of this gene are actually born with cleft palates, and that's actually one of the phenotypes you often see with individuals with this deletion, like you do with many of the different CMVs. You get these kind of sort of other abnormalities. And there's this paper that was published by Caroline Relton's lab, was it last year, a couple of years ago, and they actually did an EWAS of orofacial, like cleft palate and things like that, and this gene actually came up as one of the top hits. So maybe this particular region is picking up the cleft palate, which also gives us kind of more support that what we're doing is right. Can we pick these things up, then maybe we can also pick sort of denomethylation signals that might be to do with the neuropsychiatric phenotype of this disorder as well as the actual physical. And then this is the other model we ran. So here we were just looking to see within the actual 22q11 carriers. So obviously we had a much smaller number here. And so we had, so we're looking at basically 37 with psychosis and 27 without, and to see if there was anything different between the individuals with the deletion that had psychosis and those that didn't. We didn't find any probe that reached significance, genome-wide significance, but that's probably because we have quite small numbers for this analysis. But we did do a regional analysis, and we did find two regions, these two genes here, NBR2 and RUFY1, and both of which have been associated with schizophrenia before or bipolar disorder. The RUFY1 has been differential expression in messenger RNA has been found in the cortex of bipolar patients, and there's also evidence suggests that NBR2 is differentially expressed in schizophrenia. So we then did a network analysis using this software called WGCNA. This is actually software that has been designed to use with gene expression data. So you have to, because we get so many probes from the EPIC array, we have to sort of skim down our data set a little bit, so we were able to use this software. Otherwise it just crashes our server. So what we do is we just take the most variable probes and then look to see if we can see any discrete modules of co-methylation. We had quite a lot of different variables with this data set as well. We had different sort of biochemical, particularly just within the 22q11 deletion patients. One of which was the ratio of urinary dopamine with the urinary homovanillic acid, which are both metabolites of dopamine, and we found a significant module that was co-methylated with this measure, and another one was with cleft lip. So this is just the kind of map you get. Obviously, you can't see that, but it gives you an idea of the different variables we had with this data set. So then we could plot this one particular module that was associated with the dopamine metabolites out, and you find the genes that are in the hub, so they're the most connected